Welcome to the Explainer. So today we're taking something that honestly usually feels super intimidating and, well, a bit too academic, and we're going to completely demystify it. We're looking at quantitative nursing research, but we aren't going to look at it like some dry textbook. I want you to think of this as your personal guide to conducting a high-stakes, real-world medical investigation, because ultimately that's exactly what clinical research is. It's detective work where the clues are numbers, the suspects are variables, and solving the case can literally save lives.
Okay, let's dive right into this. Here's our roadmap for today. We'll start with why nursing research actually matters, define what quantitative research even is, and check out the different types of studies. Then we'll break down variables and hypotheses, look at how we process data, and wrap up with the statistical tools and best practices.
All right, Section 1, Why Nursing Research Matters. Before we get bogged down in the heavy data, we've got to remember why we even do this. I absolutely love this quote from our source material. Research requires courage. It is done by experts. You know, good research isn't just about being systematic and empirical, though sure, it definitely is those things. It takes genuine courage to question the status quo in medicine, right? It takes serious guts to look at a deeply ingrained hospital protocol and say, wait, is this actually working, or can we do better? That right there, that's the true heart of a medical investigator.
And the real-world impact of that courage is pretty incredible. Think about this chain of events. It literally all starts with research. That research generates brand new knowledge and new technology. But it doesn't stop there. That new knowledge leads to completely new clinical behaviors, and that technology leads to new clinical practices. And what sits at the very end of this whole chain? Improved quality of life. The numbers you're crunching out there directly result in better outcomes for the actual patient in the bed.
Moving right along to Section 2, what is quantitative research?
So if research is our medical investigation, quantitative research is essentially our forensic toolkit. Let's break these traits down a bit. The academic jargon says it's associated with the positivist tradition. Well, all that really means is we believe there's an objective reality out there, and we can actually study it and know it. How do we do that? By gathering hard numerical values. We use deductive reasoning, meaning we move from broad general theories down to specific observations. And absolutely crucial here, we impose strict control. We want to completely minimize human bias so that the empirical evidence, those hard numbers we collect in a natural or lab setting, tells us the absolute truth about whatever concept or variable we're investigating.
Which brings us to Section 3, Types of Quantitative Studies.
The absolute most foundational level here is descriptive research. This is where you're essentially mapping out the scene of the investigation. Descriptive research answers the basic, crucial questions, the who, what, when, where, and how of a situation. You aren't manipulating anything here at all. You're just describing a given state of affairs. So, for example, what are the exact characteristics of a specific patient group? Or what are the smoking habits of healthcare providers in a certain hospital? You're just establishing the baseline facts.
Next up, we step into correlational research. This is where our detective work starts looking for relationships between variables. We're trying to figure out if and exactly to what degree a mathematical relationship exists between two things. But, and any good researcher knows this, we have to remember the golden rule. Correlation does not equal causation. Just because two variables happen to move together doesn't mean one is actually causing the other. It implies we might be able to predict something, sure, but we need way tighter controls to prove true cause.
And that brings us to the big one, experimental research. This is literally the pinnacle of quantitative proof. It's where we finally get to study true cause and effect under highly controlled conditions. Basically, we introduce an intervention to an experimental group, hold back on a control group, and then measure the outcome. A great example from our source material is testing the effects of verbal suggestion on the overt pain reactions of postoperative patients. We strictly control the environment just to see if our specific intervention directly causes a change.
All right, Section 4, Variables and Hypotheses Explained. To really wrap our heads around this, let's anchor it with a real-world nursing problem, skin breakdown, or bed sores in comatose patients. Now, what's really interesting here is how cleanly the concept splits our problem into cause and effect. The independent variable is the cause. It's the thing that we as researchers actually introduce or manipulate. In our little mystery here, this is the presence of a specialized airbed. The dependent variable? That's the measurable outcome, the effect we're watching for. Here, that's the incidence of skin breakdown. Ultimately, we just want to know, does changing the independent variable actually change the dependent variable?
To answer that, we make an educated guess, which we call a hypothesis. But in research, we usually look at two competing guesses. On one side, you've got the null hypothesis. Think of this as the ultimate skeptic. It assumes there is absolutely zero relationship between our variables. It states that airbeds have no effect whatsoever on skin breakdown. On the flip side, we have our directional hypothesis. This is our bold prediction. It doesn't just vaguely say, hey, there's a relationship. It predicts exactly what's going to happen. Comatose patients placed on an airbed will have a lower incidence of skin breakdown than those who aren't. Our job as the investigator is to crunch the numbers and see exactly which hypothesis survives.
Moving on to Section 5, Processing and Analyzing Data.
So once we run our experiment, we transition from the what to the how. Researchers follow a super logical four-step sequence to transform raw, messy clinical evidence into something we can actually read. First, editing, basically cleaning up the raw data. Second, coding, assigning numbers or specific categories to the responses. Third, encoding, actually entering all this into a data file or software program. And finally, tabulation, generating those neat, readable data summaries, trends, and patterns. It's essentially taking a mountain of chaotic paperwork and turning it into organized truth.
But, the type of analysis we can actually pull off depends entirely on our scale of measurement, which becomes increasingly precise as we go. At the most basic level, we've got nominal data. These are just categories without any real math values, like biological sex or hair color. Step up from that, and we have ordinal data, categories you can rank, like a degree of malnutrition, but the mathematical distance between those ranks isn't exactly equal. Next is interval data, where the numerical distances are equal, but the 0 point is kind of arbitrary. Think of body temperature in Fahrenheit. 0 doesn't mean a total absence of heat, right? Finally, the absolute most precise is ratio data. This one has a fixed, true 0 point, like the number of children a patient has, or the hours they spent studying. Knowing your scale is absolutely crucial because it tells you exactly which statistical tool to reach for.
And finally, Section 6, Statistical Tools and Best Practices.
All right, now we're going to decode the heavy statistical tools used to finally solve our medical mystery. First up is the t-test. This is basically your go-to tool when you need to keep things straightforward. It's specifically designed to examine the statistical difference between the means of two distinct groups. Say you want to compare the blood pressure of patients on medication A versus medication B. The t-test is exactly what you'd use.
Next, we have the z-test. Keeping the pace punchy here, the z-test is actually very similar, but it's used to test for a significant difference between means from totally different and totally unrelated populations. You'd usually bust this out when you have a really large sample size and you already know the population variance.
But what if your investigation involves more than just two suspects? Well, that is where analysis of variance, or ANOVA, comes in. You can think of ANOVA as the more powerful, versatile cousin to the t-test. It's an inferential statistic that lets us compare the means of 3, 4, or even more groups simultaneously, all without increasing our margin of error.
Wrapping up this rapid stats overview, we've got Pearson's. If you remember our correlational research from a bit earlier, Pearson's is the mathematical tool we use to actually verify it. It's a pretty straightforward test to identify if two variables are mathematically linked. Like, if one goes up, does the other go up too? Pearson's is what gives you that hard numerical proof.
So, the crucial point here is this. Doing this kind of work definitely requires discipline, but you don't have to do it alone. To survive and really thrive in quantitative research, you've got to follow some best practices. Always use a technical format as your guide, and definitely keep a copy of your research agenda close by. Also, this is a huge one. Have a resident statistician. Seriously, you do not have to be a math genius if you've got an expert on your team. Use your checklists, learn by asking questions without any fear, and most importantly, ensure your final conclusion directly addresses your main research title. Don't lose the plot of your own investigation midway through.
We've gone all the way from the philosophy of why research improves lives, right down to the nitty-gritty statistical tools used to actually prove it. Quantitative research gives you the incredible power to find objective truth in healthcare. So I want to conclude by turning the investigation over to you. Looking around your own ward, or maybe your own studies, what pressing nursing problem would you want to solve with your own clinical research? The tools are literally right here, waiting for you. Get out there and start investigating. Thanks so much for joining me on this explainer.